Hi everyone, we are Group 4. Today we will be presenting a paper entitled Clinical Utility of Targeted RNA Sequencing in Cancer Molecular Diagnostics, published in Nature Medicine. My name is Josh, along with my co-presenters Maliga and Anwisha, and we will walk you through this large-scale clinical study evaluating how shifting our diagnostic focus from DNA alone to integrating targeted RNA sequencing can change how we detect, classify, and ultimately treat complex malignancies. So, what exactly is targeted RNA sequencing and why was this chosen for this paper? So, unlike whole transcriptome sequencing, which sequences all RNA molecules in a sample, targeted RNA sequencing selectively enriches and sequences genes or transcripts that are clinically relevant to cancer. This approach offers several advantages, such as lowering costs because only genes of interest are sequenced, higher sensitivity since sequencing depth is concentrated on clinically important targets, and analysis becomes more streamlined and clinically practical. Importantly, targeted RNA sequencing also performs well on compromised samples, especially formalin fixed paraffin embedded or FFP tissues. However, there's an important challenge. While DNA remains relatively stable in FFP tissue blocks, RNA is prone to degradation. Therefore, maintaining RNA quality becomes an essential aspect of successful targeted RNA sequencing. In this study, the authors utilized Illumina sequencing by synthesis technology to generate cDNA libraries from extracted RNA. So, now, why choosing the right sequencing method matters is because different cancers are driven by different types of genetic alterations, and each sequencing approach captures different layers of this variation. To understand this, we first need to look at the main types of genetic alterations that occur in cancer. The first are single nucleotide variants or SNVs, where a single nucleotide in DNA is substituted. Second, are insertions and deletions, commonly referred to as indels, which involve small additions or losses of nucleotides. However, an especially important category in cancer is gene fusion. Gene fusion occurs when parts of two different genes become abnormally joined due to structural genomic rearrangements, often producing abnormal fusion transcripts or proteins that can act as strong oncogenic drivers. This is where RNA sequencing becomes particularly valuable. DNA sequencing can sometimes struggle to detect fusions because of genetic breakpoints often occur within large intron regions that are difficult to sequence effectively. In contrast, RNA sequencing directly captures expressed fusion transcripts, making detection much more sensitive and specific. This is clinically important because approximately 16% of adult cancers and 39% of pediatric tumors contain large fusion events. So to evaluate this approach clinically, the study analyzed a large and diverse patient cohort. A total of 2,310 patient samples were tested, with a median patient age of 13 years old. The cohort includes both in-house patients and externally referred samples, showing that this diagnostic workflow can be immediately implemented across multiple institutions. Importantly, sequencing success was extremely high at 99.6%, despite the fact that most samples came from FFP tissues, which again is generally considered challenging for RNA preservation. Among central nervous system and solid tumors, approximately 89% of the samples were from FFP, while leukemia samples mainly consisted of bone marrow aspirates and peripheral blood. The low failure rate demonstrates that targeted RNA sequencing is practical not only in ideal laboratory conditions but also in real-world clinical environment using routine pathological sampling. So the process begins in the wet lab with the RNA extraction from tumor samples. Following extraction, RNA undergoes library preparation using the TrueSight RNA pan-cancer panel, which enriches clinically relevant transcripts. Sequencing is then performed in Illumina, MiSeq, and NextSeq instruments, generating paired end reads with a median sequencing depth of around 10 million reads per sample. Once sequencing is completed, the process transitions into dry lab bioinformatics. First, sequencing reads are aligned to the reference genome which is the human genome build HG19 using the star. Next, duplicate SNV and intels are identified using variant calling software such as Strelka and annotated using the DBSNP, GenomeAd, ClinVar, and Cosmic database to determine their clinical relevance and population frequency. To separate true oncogenic drivers from genetic background noise, row variants were filtered and subjected to a strict quality floor of 10% VAF, 20 read minimum depth, and at least 5 mutant reads. The workflow also includes fusion detection using metafusion, and gene expression analysis was done using RSM and Combat SEQ. Finally, important findings undergo validation through orthogonal methods such as DNA sequencing, qPCR, IHC, or IHC to ensure clinical reliability.
So, following that workflow, the current study aimed to evaluate the ability of target RNA sequencing in the cancer treatment. We considered the following aspects. Let's see one by one. At first, we measure the RNA expression level, then we normalize those data to log 2 TPM plus 1 scale. Then this step is very important because it helps us to cut down the noise. Also, it will tell us the true copy number variance. In other words, it will tell us the total number of reads. On this side, you can see oncogenic genes. On the other side, you can see tumor suppressor genes. On the y-axis, we have the normalized RNA expression levels. The results suggest that oncogenic genes are overexpressed and the tumor suppressor gene is underexpressed. So from this we can conclude that target RNA sequencing helps us to predict simultaneously the oncogenic gene levels and the tumor suppressor gene levels. This data is considered as very important for the cancer treatment. Although based on the RNA expression levels, we cannot conclude anything. So it is very important to know what caused this genetic alteration. This slide focuses on genetic mutations caused by those genetic alterations. The workflow is very simple. We extract the RNA, then we convert it into readable format. We sequence it, align it, and map it to the reference genomic data. It will tell us variant, and variant will tell us variant is nothing but small changes in the genetic code. But in the case of tumor cells, we have thousands of these variants, so it is very hard to interrupt them. So the author used dual confidence filtering system to map those meaningful mutations and summarize those data in the heat map. The heat map column represents the patient samples and row represents the mutation caused by those genetic alterations, and different color represents the intensity of those genetic alterations. In here, the red represents the genetic alterations with biological significance that drive the tumor growth, and the blue represents the variants that may be sometimes noise. It need to be carefully studied. So by organizing the data this way, it will help the doctors to easily differentiate between what is noise and what is signal. So now we know the mutations, but we don't know what caused these mutations. So after identifying the splice site mutations through the variant calling, the same aligned RNA sequence reads were analyzed for splice junctions. The software detects these abnormal exon-exon connections. Here we have shown the two classical examples, TP53 and NF1. These are tumor suppressor genes. They help us to protect our body from the cancer. And the researchers detected that there is a mutation at the splice acceptor site and the mutation in the intron region. These mutation disturb the disturb the normal splicing and thus results in the abnormal mature mRNA. So from these results we can conclude that target RNA sequencing not only help us to detect the mutation, it also help us to detect the splice site error caused by those mutations. So from overall lab data, we can conclude that this single assay can provide multiple useful informations by using different kind of software. Although it provides valuable data, we cannot blindly depend on the lab data. So it is very necessary to prove them in the clinical. So moving on to the clinical validation, it first raises a key question: why this technique? It's because 80% of the lab data contain clinically relevant information. How is it possible? How do they have this clinically relevant information? Because of the assay. This single assay can provide or tell us a wide range of genetic alterations. Panel C, D, and E talk about the tumor biology across different tumor types. A patient carries +1 to 2 plus oncogenic variants. And panel F and G talk about the correlation between the DNA mutations and the RNA mutations, and this tells us that the target RNA sequencing test is more accurate. Although it is accurate, it is considered only valuable if it helps to treat the real patient. So moving on to the core clinical finding of the study. As we all know that target RNA sequencing generated useful information, and those useful informations are classified into four types. The first one is targetable findings. These help us to treat the patients with drugs, and the second one is diagnostic finding. These help us to differentiate tumor types, and third one is non-cancerous samples. These are supportive samples. These help us to resolve the challenging cases. Panel B, D, E, and F tell about this distribution across different tumor types. CNS tumor provided most of the diagnostic information, whereas solid and hematopoietic tumor provide both diagnostic and the targetable finding information. To further ensure their reliability, the author also checked the non-cancerous samples. You could see in the panel C, this helps us to prevent the false findings. So this data is considered as core clinical finding of this study because it is directly coming from the RNA expression levels. So let's see how these data are transforming the treatment. Next, we're moving on to the clinical impact by the target RNA sequencing. Uh, the team find out that 48 tumors are reclassified after target RNA sequencing. Based on that finding, they selected 104 patients. Among that, 94 patients were treated, and most of the treated patients' tumor types are CNS and leukemia. Panel D and E talk about the pathways and the drug benefited by this targeted therapy. MAPK and tyrosine kinase dominated therapy over the other tumor types. So overall, we can conclude that target RNA sequencing not only help us to diagnose, it also help for the treatment or to make decisions for the treatment. So from the overall results, we can conclude that target RNA sequencing closes the gap between the complex lab data and the clinical treatment. Also, it helps the doctors to make right decisions at right time.
Before the clinical use, the assay was rigorously validated against seven orthogonal methods: IHC, DNA NGS, droplet digital PCR, nanostring RT-PCR, SNP RNA, SNP array, and the FISH. Results show that 98% sensitivity and 100% specificity for the SNVs or the indels, and 100% sensitivity and specificity for fusions. It showed 93.3% concordance with the DNA sequencing, and the call cutoffs are conservative. Like the VAF was more than 10%, and the minimum depth was 20 times, and with more than 5 mutant reads. The study was approved by the Sick Kids Research Ethics Board, run in a accredited lab and fully Helsinki Declaration compliant. So the RNA is critical, and specifically with the FFP samples, which made up to the 76% of cases. So strict QC parameters were applied, among which was the minimum of 50 nanogram RNA with a DV200 of more than 40%, and the median sequencing depth of 100 million reads, which is like 2 cross 75 base pair paired end on the MI-sec or the next sec. Overall failure rate was only 4.8%, which is like 110 out of 2,310 samples. Importantly, 93% of the failures were due to the insufficient RNA, not sequencing, giving a sequencing a success rate of 99.6%. These QC thresholds ensure reliable results even from the challenging clinical samples. Looking ahead, the targeted panel already maximizes yield while minimizing cost and depth. RNA sequencing also provides expression data for the immune infiltration signatures and oncogenic pathway activation, and detects expression misregulation from the amplification, deletion, epigenetic, or the transcriptional changes. Low VAF variants, such as low as 4 to 5%, are reliably detected. In 103 paired RNA DNA cases, 93.3% concordance was seen, and RNA sequence uniquely identified 2.3% of the variants, filtered 38 VUS from the DNA sequencing, and revealed allelic imbalance, which is an information that DNA alone cannot provide. The assay already supports less invasive sampling. 71% of the leukemia cases from the bone marrow aspirate, and then 29% from the peripheral blood. FFP transports also expands access to low and middle income countries. Clinically, 30% of the eligible patients received targeted therapy based on the RNA sequencing. While the therapy was actively considered, 92% received it, which was a total of 94 patients. Top therapies were MAPK inhibitors, which was 41%, tyrosine kinase inhibitors, 31%, and the immune checkpoint inhibitors, which consisted of 12%. In the summary, the study establishes targeted RNA sequencing as a powerful diagnostic tool with an 87% clinical utility across the 2,310 patients, comparable to the DNA sequencing but with added functional insight. It enhanced detection, 48 revised diagnosis, 37 fully new, with fusion found in 22% adults and 36% of pediatric cancer. Notably, 86% of fusion driven tumors had no other variants. It supports precision oncology. 94 patients received targeted therapies working across all the sample types and ages from infants to like 90-year-old people. The final takeaway is that targeted RNA sequencing is transforming cancer diagnostic. First, it dramatically improves the fusion detection, outperforming DNA sequencing because RNA captures expressed fusions directly. Second, it enables better treatment decisions, directly leading to targeted therapy in 94 patients, with 92% of considered patients actually treated. And third, it is becoming essential. A single asset that uses less tissue, costs less, and delivers higher yield across the FFPE, frozen, bone marrow, and blood, and all other age groups. So, with that note, I'd like to end the presentation. Thank you, everyone.